Hello, my name is Victoria. My group members are Eunice and Hannah, and we are presenting a paper titled Composition and Divergence of Coronavirus Spike Proteins and Host ACE2 Receptors Predict Potential Intermediate Host of SARS-CoV-2. The significance of this study is that SARS-CoV-2 is affecting people all over the world. Europe has experienced a large amount of cases, with Italy, in particular, experiencing a great deal of cases. Not only has Europe experienced a large amount of cases, but so has Australia. The United States and Canada are next in line for the top cases around the world. Within the United States, the Southeast, Northeast, and West Coast have experienced the most cases. There are numerous deaths that have occurred across the world due to the coronavirus. These deaths have occurred predominantly within Italy, the rest of Europe, and the United States.
Back in 2003, SARS-CoV was a respiratory epidemic that originated in southern China and infected over 8,000 people. The virus was transmitted from bats to humans through the intermediate animal palm civets, which are small weasel-like animals. MERS-CoV, which had similar symptoms and was first identified in Saudi Arabia in 2012, infected humans primarily through contact with dromedary camels. Similarly to SARS-CoV, bats are believed to have passed this to camels. With the current pandemic, SARS-CoV-2, knowing the intermediate is crucial for preventing further spread. On the surface of all coronaviruses are spike proteins. These proteins mediate the entry of the virus into the host cells. Spike proteins are glycoproteins and type 1 transmembrane proteins. They are made of anywhere from 1,100 to 1,400 amino acid residues. The spike proteins assemble into trimers on the viral surface. The protein has two main subunits. S1 contains the receptor binding domain, an important area on the protein made of about 200 residues. The receptor binding domain is key in understanding the viral origin. The S2 subunit is responsible for the fusion of the virus to host cellular membranes. The spike protein mediates entry of the virus into host cells via the ACE2 receptor protein, which is largely abundant in humans in the epithelia of the lungs and small intestine. This is a structural diagram of the spike proteins present on SARS-CoV, MERS-CoV, and SARS-CoV-2. Within the S1 subunit are the signal peptide, N-terminal domain, and the previously mentioned receptor binding domain. The S2 subunit contains the fusion peptide and heptad repeats 1 and 2. Separating the two subunits is the S cleavage site. While each of the spike proteins are slightly different, the structures are all similar.
Determining the intermediate host of the spread of SARS-CoV-2 would be beneficial to prevent further spread. The goal of the study was to identify the possible intermediate host of SARS-CoV-2. The study compares spike sequences between SARS-CoV-2 with SARS-CoV, bat SARS-like CoV, and other coronaviruses. The paper suggests that turtles, along with pangolins and snakes, may be intermediate hosts. Next, we are going to discuss the procedures in this experiment. First, the sequences of the respective proteins were retrieved. Full-length protein sequences of spike glycoproteins and ACE2 were downloaded from the NCBI GenBank database. The proteins downloaded included SARS-CoV-2 spike protein, SARS-CoV spike proteins, and bat SARS-like CoV. GIS aid was used to download the bat SARS-like CoV RATG13 sequence. NCBI Bioproject database was used to download the pangolin metagenome. The coronavirus genome sequences were analyzed by Verma. Next, the protein sequences were aligned and analyzed. MegaX was used to align spike protein sequences from different sources and to accomplish residue comparison of ACE2 among different species. The phylogenetic analysis was also completed through multiple comparisons using MegaX. Clustal W multiple sequence alignment was used to complete multiple comparisons. Finally, structure and binding model of spike receptor was made for further analysis. The iTassar server online tool was used to simulate the full-length structure of SARS-CoV-2 spike glycoprotein. PRISM 2.0 was used to predict the spike ACE2 binding model. Lastly, PyMol software was used to analyze the spike protein and ACE2 structure files.
The interaction between the receptor binding domain and the receptor is the key factor in determining the host range of the coronavirus. The model shows the interaction between the human receptor and SARS-CoV-2 spike protein. This area is the external domain of the spike protein. The amino acids of the external domain of spike protein interacts with the amino acids on the human receptor. This interaction could be different for different amino acid sequences. Keeping this in mind, we are now going to look more closely into the protein alignment and the key residues before going into the sequence analysis.
This figure shows spatial alignment between the spike proteins of SARS-CoV and SARS-CoV-2 predicted by protein threading. Protein threading is currently one of the most reliable ways to predict the 3D structure of a protein. It utilizes a template structure, in this case, the template structure is SARS-CoV, to calculate the most statistically probable structure of the protein in question, the protein in question in this case being SARS-CoV-2. The threading alignment is the number of aligned amino acid residues in the proteins divided by the entire length of the protein. A threading alignment greater than 1 is thought to be good. SARS-CoV and SARS-CoV-2 were found to have a threading alignment of 6.27, indicating that the spike proteins of the viruses were highly aligned. However, they are unique enough to be different viruses. The differences in between the spike protein structures are apparent when looking closer at the external subdomain of the protein. A closer look at the external subdomain of the receptor binding domain shows the specific amino acid residues that interact with the human ACE2 receptor. The key residues in SARS-CoV that bind to the ACE2 receptor are leucine, asparagine, and threonine, while SARS-CoV-2 uses the residues leucine, phenylalanine, glutamine, asparagine, and tyrosine. The two residues that both receptor binding domains have in common are leucine and asparagine. Leucine interacts with the ACE2 receptor using hydrophobic forces. On the other hand, asparagine has the ability to form hydrogen bonds with the ACE2 receptor. The greater amount of binding residues on SARS-CoV-2 may induce tighter binding of the virus to human ACE2, making SARS-CoV-2 more infectious.
Up to now, the binding residues of the SARS-CoV and SARS-CoV-2 at the external domain were compared to see the differences in the structure and interaction of the human ACE2 receptor. To determine the intermediate host of SARS-CoV-2, this study observed the receptor binding domain sequences from SARS-CoV, bat SARS-like-CoV, pangolin SARS-like-CoV in comparison to the sequence of SARS-CoV-2 via multiple sequence alignments. This shows the multiple sequence alignments with the respective sequences. The boxed region is part of the external domain of the protein where the key binding residues are. Looking at this region more in detail, there are deletions from residues 473 to 490 for bat SARS-like-CoV and SARS-CoV when comparing the sequences to the SARS-CoV-2. As mentioned, there are more binding residues for SARS-CoV-2 than for SARS-CoV. Similarly, the deletions in bat SARS-like-CoV sequences show that there are no evidence that bat SARS-like-CoV infects humans directly. The viruses from the bats do not infect humans naturally and may have other intermediate hosts. The alignments from residues 329 to 521 also shows similarities in these sequences. Between bat SARS-like-CoV and SARS-CoV-2, there's 89% similarity. SARS-CoV-2 and pangolin SARS-CoV has 93% similarity, and bat SARS-CoV and pangolin SARS-CoV has 85% similarity. When focusing only on the receptor binding domain sequences, pangolin SARS-CoV has a high probability of crossing host barriers and infecting humans. We've been only focusing on the receptor binding domain of the spike protein, but now let's look at human ACE2 receptor to gather more information on the intermediate host.
Here, human ACE2 is shown in gray along with spherical models of the key binding residues of human ACE2. The residues methionine, tyrosine, aspartate, arginine, and 2 lysines are on the interface of the receptor interacting with SARS-CoV-2 spike proteins. These same residues are also on the interface of ACE2 when the receptor is interacting with SARS-CoV. It has been shown that lysine 31, tyrosine 41, and lysine 353 are especially important residues as mutations of these prevent the spike protein from binding.
These key binding residues on the ACE2 protein can also give information on the intermediate host. Next, the residues from the different species are observed, especially focusing on the residues at lysine 31, tyrosine 41, and lysine 353.
Table 1 depicts the residues comparison of ACE2 among different species interacting with SARS-CoV-2 spike glycoprotein RBD. The abbreviation RBD stands for receptor binding domain. The mutated residues are highlighted as red at the corresponding positions based on human ACE2 numbering. For the residues comparison of ACE2 among different species interacting with SARS-CoV-2 spike glycoprotein RBD, snakes, turtles, pangolins, and bats were of most importance. Both snakes and turtles were found to have 6 mutations compared to human ACE2. At position 41, every species tested maintains tyrosine except bats, which is substituted by histidine. The substitution of His 353 with Lys 353 in mouse ACE2 allows SARS-CoV to infect murine cells, indicating the importance of residue Lys 353 binding with receptor. The interaction with the receptor at position 41 may possess higher affinity with ASN 501 of RBD than histidine. Using the idea of ASN 501 and RBD domain with the sites 41 and 353 of ACE2 receptor, it can be suggested that turtles and pangolins are closer to humans than bats. This suggests that turtles and pangolins may be the potential expanded host of SARS-CoV-2.
From the past studies, bats are determined to be the natural host of coronaviruses, but intermediate hosts are all different. Proposed intermediate hosts include pangolin, turtle, snake, civet, and dromedary. This study found that the sequence of pangolins and turtles are closer to sequence of SARS-CoV-2 than that of bats. Other literature state a similar conclusion. The paper from Zhang and etc. concludes that pangolins are the most probable intermediate host, and other articles say turtles. When looking at the origin, the coronavirus started spreading from Wuhan markets, and in these markets, the pangolins were illegally traded, whereas turtles were more accessible to the public. This makes turtle the most probable intermediate host, but further studies need to confirm. Moving forward, once the intermediate host is determined, the mechanism for viral infection in animal host could be better understood for control and treatment of the coronavirus. Then further studies could be done on how the binding of the SARS-CoV-2 spike protein can be blocked to stop the viral infection. All around the world, the scientists are working hard to determine more information on the new coronavirus to face this pandemic. Thank you for watching, and I hope you stay safe.